Good morning to you all. My name is James Harden, and I am a graduate student in the Immunology program. I'm looking forward to telling you a little bit about myself, my research, and my experiences here at Stanford. So I grew up on the East Coast, far from here, in Charlotte, North Carolina, and I went just 30 minutes north of there to a small liberal arts college called Davidson. And after graduating, I had realized I still had no idea what I wanted to do or where I wanted to be. Honestly, as a first-generation student, it was a little jarring for me to have so many of my peers already with multiple research experiences and many with publications. Meanwhile, I had just learned how to use a pipette. So I took a year at UNC Chapel Hill to do a postbac program in research. And this is where I realized the critical factors that was going to be important to me in choosing a grad school, in terms of the type of research I wanted to do and the type of researcher I wanted to be. Ultimately, I wanted to be in a place where my research identity and my social cultural identity could mesh together as one. So after much anxiety and deliberation, I chose Stanford Biosciences. And while there are many things I would have done differently, I will still choose Stanford all over again. I think you quickly realize the privilege of being in a place as well-funded and well-resourced as Stanford, especially when it comes to pushing forward your research. And also the benefit of being in a place that fosters a community of so many talented and supportive people that really makes the PhD experience enjoyable. Now, I love all things immunology. I find it incredibly fascinating and complex, especially because our immune system is made up of so many different cell types that all work together and individually play unique roles in protecting us. Many of us are familiar with the immune system's role in serving as a shield that protects us from pathogens. But in the context of transplantation, which is what I study, this immune response is actually a problem. There's over 100,000 people in need of a transplant every year. And our immune response actually prevents successful transplant outcomes because it attacks the transplanted organ the same way it attacks pathogens. When our immune cells sense any sort of foreign substance, including a transplanted organ, it induces an inflammatory state. During this process, these various cell types will undergo many different structural and functional changes that contribute to your overall immune response. These changes are reflected in the many different proteins that are up or down regulated on the surface and in the cytosol of the cell. And ultimately, this can lead to transplant rejection. One of our major goals, then, has been to be able to capture these dynamic changes that occur during inflammation and that is specific to transplant rejection. One way to do this is to model transplant rejection in mice, the benefit being that it allows us to study the immune system in a very controlled way. In this transplant mouse model shown here, we transplant a donor heart into a genetically identical or genetically dissimilar recipient, called a syngeneic and allogeneic transplant, respectively. We see that the model works as expected since we see no rejection in the syngeneic transplant, whereas 100% of allogeneic transplanted hearts reject by day 7 post-transplant. With this, we are trying to recapitulate the transplant rejection we see in humans. This information gives us a critical window to study the immune cells. We therefore collect multiple tissue samples at days 1, 3, and 5 post-transplant. And we isolate the immune cells from these tissue samples and perform high-dimensional cytometry analysis where we measure the expression level of more than 40 different proteins on each of these different cell types. And this basically reflects the cell's phenotype, function, and activation state after transplantation. We visualize this high-dimensional data using UMAP, which is Uniform Manifold Approximation and Projection. And this is just a fancy way of saying that when you measure the 40 different proteins on each of these cells and then graph it on a two-dimensional UMAP, what we will see is that the cells that have the most similar protein profiles will group more closely together. And indeed, when we plot our data on this UMAP, we get a global overview of the different cell populations that make up our immune system, where every dot on this plot represents a cell that is color-coded by the cell population it belongs to. With this data, we can then partition it by condition, because what we care about is whether or not we can identify a cellular profile that is distinct in transplant rejection. The dots shown in dark blue on the left are the cells from the syngeneic transplant, while the dark red on your right are the cells from the allogeneic transplant. The gray background is just the scaffold of the original map. At day 3 post-transplant, shown here, we see that the two conditions look very similar. But by day 5, we can appreciate how very distinct the allogeneic transplant looks, shown on your bottom right. There are multiple areas of this UMAP that show disparate spatial distribution of immune cells, indicating a systemic shift in protein expression across multiple cell types. This essentially tells us that there are multiple clusters of cells responding to the transplanted organ. looking across these different cell types, we can appreciate the moderate differences within the T cell and NK cell compartments, and the rather drastic differences that we can identify in the myeloid cell compartment. This is showing a clear phenotypic signature of rejection that is identifiable by day 5 post-transplant. So the next step, we want to characterize the specific cells that are reacting to the allogeneic transplant. And to do this, we select just the cells within the circle here and recluster them. I'm highlighting for this presentation the myeloid cell compartment as an example. As shown in the middle, we get higher granularity with 12 new clusters representing various myeloid cell populations that we can identify. And as shown on the far right, we see one particular cluster, starred and shown in red, that is highly predominant in rejection. Importantly, when we quantify the proportions of these cell clusters, we see that cluster 1, shown in red, makes up about 50% of all myeloid cells in allogeneic day 5, and is low frequency in every other condition. This is a drastic phenotype of rejection. To characterize this cluster, we then identify the 8 different proteins that distinguish this group of cells from all the other clusters. And this information will be useful in order to identify these cells for downstream analysis. So it's cool that we can identify such a distinct signature of rejection by day 5, but we wanted to see how robust the signature was. So we compared our heart transplant model to a completely different limb transplant model involving the skin, bone, muscle, and other tissue types. And what we find in this principal component analysis is that this signature is relatively conserved across both transplant types. Each shape represents one mouse transplant, color-coded by syngeneic or allogeneic. The circles represent heart transplants, and the triangles represent limb transplants. This conserved signature is evident by the fact that the light red events, representing the allogeneic at day 5, clusters together regardless of whether or not it is a heart or limb transplant. So in the grand scheme of things, we hope to use this data in characterizing the immune response in rejection to be able to identify predictive biomarkers that lets us know if a rejection will occur before it actually happens. And additionally, we want to be able to develop immune suppressive therapies that specifically target allogeneic responsive cells in order to improve transplant outcomes. So the PhD journey is not without its challenges. This graphic from a fellow grad student sums up the process nicely, showcasing the many ups and downs we all will experience along the way. This, of course, is why the decision you make for grad school is so incredibly important. So I'll share with you the best advice I received. 6 years ago today, when I was in your position, I remember Assistant Vice Provost Anika Green sharing with us what ended up holding true for me all these years. She looked at us dead in the face and said, "You must find your hype crew." The people in your hype crew are going to be the ones that will help sustain you through the trials and tribulations you will face in grad school. And your hype crew will come in many different forms, as illustrated here. And I can truly say that Stanford understands the importance of building a community for its students, with the many initiatives and resources that are here, combined with truly caring administration and staff. And my hype crew at Stanford is extensive across multiple fields, disciplines, and programs at Stanford. And even when the pandemic tries to hold us down, I have had the incredible opportunity of working with and seeing on multiple occasions how the Biosciences administrative team have put out fires, held us together, redesigned curricula from scratch, and have holistically supported me throughout this whole process. And shown here is an outreach program that I have been heavily involved with as just one of many examples of the biosciences community rallying together. So from my personal experience, I know that whether it be on campus or on Zoom, the Stanford Biosciences team is driven, engaged, and wholeheartedly dedicated to providing you the most rewarding and enriching grad school experience where you can build both your research and your community. So with that, I want to commend you all for trailblazing through these unpredictable times and being the high achievers that you are. I will leave you with my contact in case you have any questions. And it has been my pleasure to be able to speak to you all today. And I wish you the best moving forward.